Hello and welcome to IB Bio 1.4 notes. So first we'll talk about passive transport. So there's three types of passive transport. We have simple diffusion, facilitated diffusion, and osmosis. So diffusion is the movement of particles from a region of high concentration to a region of lower concentration. So as we see here, from higher to lower concentration, and is the result of the random motion of particles. So, as I said before, there's two types of diffusion, simple and facilitated, and they're both affected by the following four factors. So, temperature. The greater the temperature, the greater the kinetic energy of particles, and therefore the greater the rate of diffusion. The surface area. So, obviously, the greater the surface area, the more space there is for diffusion to occur. So, the faster the rate of diffusion. And we have the size of particles. So the smaller the particles, the faster the rate of diffusion. And finally, the concentration difference. So basically, the larger the difference between the higher concentration and the lower concentration, the bigger and faster the rate of diffusion. So simple diffusion only occurs in gases and liquids, and it requires a concentration gradient, as I said before. Then some particles cannot diffuse through the plasma membrane since they're too big or have a charge, meaning they're polar, so they don't fit through the hydrophobic tails, they're repelled by them. So in these cases, a protein would be needed, and that is facilitated diffusion across the membrane. So it could be either a channel or carrier protein. And then osmosis, as I said, it is the passive transport of water molecules from a region of lower solute concentration to a region of higher solute concentration. So from high water potential to low water potential across a permeable membrane. So this basically means it moves from an area of lower solute. So we could use salt as an example. It moved from an area of lower salt to an area of higher salt to balance out the solute concentration. So this is really important in medical procedures. So for example, in transplant operations, tissues need to be kept in a saline solution. So this is because it's important that the osmolarity, which is the measure of the solute concentration of the saline solution, is the same as that in the cytoplasm to prevent any osmosis, so the gain or loss of water through the membrane of the cell, as that would damage the tissue. So, for example, if there's too much gain of water, the cell would swell and burst, and if there's too much loss of water, it will shrivel and die. And we have three types of solutions. So hypertonic solutions are solutions with a higher concentration of solutes than water. Hypotonic solution is a solution with higher concentrations of water than there is solutes. And isotonic solution means equal solutions of both water and solutes, and an equal concentration of solutes on both sides. So this is important and confusing as well. If you see on a test that a cell is put in a hypertonic solution, this implies that the solution outside the cell is hypertonic, which also means that relative to the cell inside would be hypotonic relative to this solution outside. And then active transport. So this is similar to passive transport, except it requires ATP, energy. So we said passive and facilitated diffusion do not require energy, but active transport does. And it requires it since it moves against the concentration gradient. So an example of this could be seen in potassium ion pumps. So these pumps are important in the nervous system for the maintenance of resting potential in neurons. So basically, the concentration of sodium Na ions need to be higher outside the neuron, which is right here, than inside the neuron, while potassium ions should be more concentrated inside the neuron right here than outside the neuron. So the process for the maintaining of this gradient can be described in the following stages. So when the pump is open to the inside of the cell, right here, 3 sodium ions, you see them right here, enter the pump. this protein and attach to their binding sites. And then ATP donates a phosphate group, as we see right here, to the pump, and this causes the protein to change the shape and expel the Na plus ions to the outside. And then two potassium ions from the outside then enter and attach to their binding sites right here. And the binding of the potassium ions lead to the release of the phosphate right here. So the phosphate leaves, which causes the pump to change shape again so that it's only open to the inside of the cell. And then the potassium is, of course, released inside. And the process can be repeated. So we see here that the end point and the starting point are the same. Then we have endocytosis and exocytosis. So endocytosis occurs when bulk and bigger quantities of substances are needed to transport inside the cell, which would not work through passive and active transport. So an example would be endocytosis, which is a process where cells take in substances from the outside by internalizing within a vesicle and then being engulfed by the cell membrane. So there's two types, phagocytosis and pinocytosis. So we see here that the substances moves towards the membrane and the membrane sort of engulfs it and breaks apart to form a vesicle. So pinocytosis is the taking in of liquid substances, as we see here, and phagocytosis is the taking of solid substances. Then we have exocytosis, which is the opposite of endocytosis. It's the release of waste outside the cell using a vesicle, and it's also divided into two types, excretion and secretion. So excretion after phagocytosis, any undigested remains of the microbe that are not useful are excreted outside the cell. So it's the complete opposite of endocytosis, as we see here. The second type would be secretion, which is, unlike excretion, it's only for proteins. So proteins that are produced on the ER, rough ER, are packaged and placed in a vesicle, sort of like this, which transports the protein to the membrane right here and binds with it to release the proteins outside. And this is all possible thanks to vesicles, which are essential in both endocytosis and exocytosis, as they transport substances to and from the membrane right here and bind with the membrane in order to eject or take in material. That is it for topic 1.4. Thank you for listening.